Good morning, everyone. All right. My name is Yvonne, and I work for Medicinal Genomics. Not only do I hand out drink tickets, but I also manage the R&D team. So, plating. Plating methods are considered to be the gold standard. The petri dish was invented in 1887. And the phone, 1887. This is the phone in This is the phone in 1887. Is this not working? This is the cell phone today. Now, if I told you all to take your cell phones away and you can only rely on the newspaper for your information, you would think that I'm crazy. Yet, we rely on petri dishes to give us results for microbial testing, and we think that qPCR or molecular methods aren't as good. Now, I beg to differ. You're growing on a plate. How do you know what it is? Unless you're a trained mycologist who has years of training, it's very difficult to determine what's growing on your plate. And even if you do know what's growing on your plate, there are things that look very similar. For instance, Aspergillus niger. Aspergillus niger has also been mistaken for Aspergillus braziliensis, and they were once deemed the same. But through molecular techniques, they've determined that they are actually different species. So why are we still doing this? You know, let's bring it into the 21st century. We have the ability, we have the technology. Why are we still relying on a standard that's 135 years old? For instance, salmonella testing. You can target portions of the salmonella genome and run molecular methods and determine that it's salmonella within as little of a 16-hour incubation. Yet if you were to confirm salmonella on plating methods, that could take up to 3 days. And then they want you, at the end of 3 days of plating and confirmation, to send that sample back for sequencing to confirm that your 3 days of plating was right. Why are we doing this? And there are some state regulators out there that aren't allowing molecular methods for species-specific detection. It's crazy, right? Why are we doing this? Why are we making it more difficult than it needs to be? So we've been publishing on these problems since 2015. The past 8 years, this is what I do on a daily basis. Try to explain and educate people why plating methods aren't the gold standard that everybody thinks that they are. Most recently, we published this past February, and I'm going to touch upon some of that data in a bit. I want to go back to a study that we did last year with Michigan and trying to determine total yeast and mold. That's another problem, totals, but we can get into that later. So there was sample, it was collected, it was homogenized by a third-party lab and distributed. It was broken up into high, medium, and low categories, and it was plated on 3 different types of plates. These are the 3 different types of plates that were used in this study, and as you can see, these 3 particular plates are supposed to be isolating yeast, mold, fungi. Yet when you look at these plates, you see very different results. The top row is DRBC plating, and you can see there's a lot fewer colonies growing on these plates than at the bottom, which is potato dextrose agar. Now, to a lab technician that doesn't know the difference between a bacterial colony and a yeast and mold colony, you're gonna get very different results. The PDA, the blue dots that you see, are counts. The majority of those blue dots that are being counted for total yeast and mold are actually bacterial. This is some sequencing that we had done. We picked colonies from these plates and we put them through genomic sequencing to see what they actually were. And on the 3 plates that are supposed to be growing yeast, mold, and fungi, you see that bacteria is indeed growing on these plates. And what's really interesting is, can you advance what? There should be a circle that comes up. Can I do that? Yes, sorry. So it's really difficult to see what that circle up at the top says, but it's a actual bacteria by the name of Total Aerobic. Now, Pantoa is a gram-negative, which makes it an entero, lactose-fermenting bacteria. Lactose fermenting makes it a coliform. So we got a bacteria, it's an entero, and it's also a coliform. And it has actually recently been, in 2019, there was a publication as a growth promoter for cannabis. Okay, so now we have a bacteria that is used to help grow cannabis. It's also been known to show skin infections in humans. So you could have this growth-promoting bacteria on your cannabis plants, and then you have your trimmers come and trim it up, and they get skin infections. What's going on? So we have done previous studies on different microbiomes, and we found that in 30 of the 172 microbiomes that we've surveyed, Pantoa agglomerans has come up. So this was interesting. So we decided to look at this with an actual ATCC culture. Just to touch back. So I said it was a bacteria, it's an entero, it's a coliform, therefore it's a BTGN. These are the different states that have regulations for those tests. I believe the top, this is states that have a total aerobic regulation. These are states that have a coliform. These are states that have an entero, and I don't know why it's not showing. And this one over here has BTGN requirement. So that's important. So we take this Pantoa agglomerans and we incubate it. And we put it on plates. And for this particular plate, this is a total aerobic count plate. It's a rapid total aerobic count plate. As you can see with the manufacturer's instructions, it says to grow it at 35 degrees, plus or minus 2 degrees, for 24 hours, plus or minus 2 hours. And when you do that at the recommended growth temperature, you can see at the bottom, there's no growth. Yet, if you grow it at 30 degrees, or you grow it at 26 degrees, you do have growth. So if labs are following the recommended manufacturer's documentation for this, which they haven't tested it in a cannabis matrix, which just said was a very important factor, it is, it needs to be tested in cannabis matrix, you get very different results. And I said that it was also an enterobacteria. Well, we grow it on the entero plates at the recommended temperature and at 2 differing temperatures, and you get no growth. Coliform plates. I said it was also a coliform. This is what happens when you plate it on a coliform plate. 26 degrees, you see it. 30 degrees, you see it. 36 degrees, it's there, but it's minimal. So if you're having labs test for these organisms at the recommended temperatures and they're not showing up, they're passing it. This isn't a fluke. This is another organism. This is Aromonas hydrophila. You see the same thing. And this is a bacteria that causes gastroenteritis in 13% of humans. Yet when you do qPCR methods or molecular methods, temperature doesn't matter. And you detect it across the assays. So again, why are we relying on a gold standard that is missing stuff? And then molecular methods, well, why doesn't it match? It doesn't match because you're comparing 2 totally different technologies. So we did another experiment, and this was in matrix. This is a hemp sample. And we decided to test this hemp sample, divide 10 grams, 3 different bags, test it at 3 different temperatures, 3 different plating types, 3 different scientists processing. So with the qPCR method, there's no incubation up front for these particular tests. For species specific, that's different. I'm not gonna get into that right now. So we did initial time point, and then we incubated those bags for 24 hours. No incubation, 3 temperatures, which weren't really put at their temperature yet. So this is showing, why doesn't it like, okay, I don't know why it doesn't show up over there, but. So 3 bags, 3 scientists, pretty similar results. This green one over here is an outlier. Unfortunately, that's me, but what are you gonna do? So the average CFU for these tests right here is 150,000 CFUs. If that was a total aerobic test, that would fail. If that was an entero, that would fail. If it was a coliform, it would fail. You incubate those at their respected temperatures for 24 hours, it goes from 150,000 CFUs to over 6 million CFUs. Clearly, what's on that plant, on that flower, is alive and growing. Yet, when you plate it, 24 hours, 3 temperatures, it would pass. 48 hours, all of a sudden, you start seeing stuff. And 72 hours, it's essentially the same as the 48, they're just a little larger. Okay, that's 1 carbon source. You change your carbon source, totally different results. This is 2xYT agar. 26 degrees is where you're seeing that growth. You're not seeing at 30 and you're not seeing at 36. This is LB agar. Again, similar types of results. So what's going on here? Well, there's a couple things. Not all organisms grow at the same rate, at the same temperature, which I think I clearly demonstrated to you before. Not all organisms grow on the same carbon source. And not all organisms grow at the same rate. And these 3 things are very important when you're dealing with plating. And this is not something that we have when we're testing with molecular methods. Now, it's very interesting. Remediation practices are becoming more common. And I think a lot of what may be happening here, and this is hypothesis, and this is something that I want to investigate further, and maybe I can present to you guys next year on my findings. But I think a lot of what may be happening here is this viable but non-culturable event. And what happens is that some of these remediation techniques involve sterilization, whether you're using ozone, gamma radiation, X-ray radiation, whatever hydrogen peroxide treatment. And they're knocking the organisms down. But it's kind of like a boxing match, as I'll use Zach terminology here. They're knocking them down, but they're not knocking them down for the count. They're down at the 8 count, they're kind of staggering back to life, and then they'll start growing again. And that could be why on some of these plates, at 24 hours, it's just not enough. But if you give it a few more days, they start reviving. And this makes sense. I mean, you kind of see it in food, right? You go to the grocery store, you buy a pack of strawberries, they look fine. You bring them home, you stick them in the refrigerator, 2 days later you go to eat them and you're like, oh shit, they're all moldy. Same thing could be happening. So just because a sample leaves the lab on day 1 and passes microbial doesn't mean it's gonna go sit on a shelf in a dispensary and not still continue to grow the mold or bacteria. So this is something that's really interesting. I think it's something that's important. And the whole point of it is that qPCR or molecular methods are gonna pick up these viable but non-culturable. And so we may be seeing, well, how come if I use qPCR, I'm failing my product, but I plate it and there's nothing there. I believe that it's this viable but non-culturable event that's happening. So am I really that early? I think I've made up my time. All right, so this is our team, staff. I want to thank these people. We're always looking for things to test, and this whole plating versus qPCR or molecular methods isn't going away, and I will continue to come here and educate you all. So if anyone has any questions, I'd be happy to answer them.